Okay, hello all. Today or this week in lab, we'll be looking at plant stomata, and we're going to be investigating transpiration and gas exchange. Um, so to just back it up a little bit before I go into what exactly we'll be doing in lab, I just wanted to point out that um, transpiration, or remind you all that transpiration is the process of water movement throughout a plant. And so you can see that I've highlighted it red here. And so water will enter through the roots, move up the body of the plant, and then exit through the leaves. And this is just a small process of the water cycle, small but important process of the water cycle. The structure that allows plants to release water into the atmosphere is stomata. And so that's what we're going to be looking at today. And so from the packet, you should be able to answer these three questions. Where are they located? What is their structure? And what is their function? We'll go over this just to make sure that you have it down for the lab. And so they are located on the epidermal surface of the leaf. So that's why we're doing epidermis peels today. Their structure is two guard cells that control the opening of the stomatal pore, and so I'll point that out here. And so the green thing that you're looking at on this slide is the guard cells. You can't quite see the division, but there's a division at the top and at the bottom of the stomatal pore. And so and so we have two guard cells. The small sliver of black that you're seeing is the stomatal pore. And so these either fill with water to open, or they release their water and close the stomatal pore. And so this stomata that I'm looking at right here, or that my cursor is moving around, is a closed cell. And the structure and the function of these stomata is that it's the site of gas exchange. So when the stomatal pore is open, CO2 can come in and O2 can come out. But there's a trade-off that happens. And so when the pores are open, they take in CO2 and release O2, but something else is lost in the process as well. And water is what is also lost. And so the question that I like to ask students, and so this is just a diagram of how the water enters the roots, travels up through the xylem tubes, and then evaporates out through the leaf. And so plants deal with this issue of losing water because a plant doesn't want to lose all of its water at once, because then it'll wilt and die. And so plants deal with this by adjusting the size and number of stomatal pores that they have. And so a few other important notes about stomata is that the plant hormone ABA stimulates the closure of stomata as well. And so ABA is a stress hormone, and this stress hormone will stimulate the closure of the stomata in response to like something like drought. And so if there's not a lot of water in the area, or they're not getting a lot of rain, they will close their stomata so they don't lose more and more water and end up dying. And so we'll go into turgor pressure and how it also mediates the opening and closing of stomata. But first, I want to talk about activity 1 because it goes hand in hand with the transpiration and what we just talked about. And so today, to investigate transpiration and what factors might affect it, we're going to be using geranium and their leaves. And so here I've drawn a nice diagram of the geranium leaf. You'll be taking the geranium leaf and petiole and basically, and sticking them in vials filled with water. And so this idea is basically that you're simulating 4 different plants. And so the water in the vial will act as the water source. And the idea is that water will travel up through the petiole and out through the leaves based on what environment it's in. And so there will be 4 environments that you're going to be looking at, or 4 different treatments. And the 4 different treatments are light. You'll be placing it in a dark environment. You'll be placing it in a windy environment, and you will also be placing it in ABA, which is our stress hormone. And so you'll be asked to make hypotheses to what will happen when you put these leaves in these different treatments. And it's important that you write these hypotheses regarding the transpiration rate and the weight of the vial. And so the way that we're going to be measuring the transpiration rate is by the weight of each of the vials. And so you'll weigh these at the beginning of the experiment, and then every 15 minutes, you'll weigh it again to figure out how much water is leaving the vial via the leaves or via transpiration. Read your packet if that was confusing. If not, we will help you in lab. It's also important to note, that 1 microliter of water is equivalent to 1 milligram of H2O. And so this is why we're doing the weight, is because we can convert it to the amount of water that has left the vial. Okay, so that is activity 1. And so going back to this, to turgor pressure and how it also mediates the opening and closing of stomata, which you guys have talked about in lab, or I'm sorry, we've talked about in lecture so far, but I thought I would just bring it back up just to remind you all. And so the opening and closing of the stomata is mediated by this potassium ion gradient. And so in a closed cell that is going to open, the solute, the potassium ions on the outside of the cell are going to be less than the inside of the cell, so they will travel inside of the guard cells, and this will also trigger the water to enter the guard cells and to try to balance those concentrations. And this is when we consider the cell to be turgid. And so this is an open stomata. And so that's how it goes from closed to open. However, when they're going to close again, the potassiums or any solute, I say any because we're using sucrose in lab. So the potassium ions are pumped out of the guard cells. That ion concentration decreases in the cells, and then the water will flow out as well to try to balance those concentrations. Or yes, they're released from the guard cells, and that's stomata that we consider flaccid or closed. So it's good to remind yourself why or how stomata open and close, because in the second part of lab, we're going to be artificially increasing or decreasing the solute concentration outside of the guard cell. And so try to think about what would happen if we artificially increased that potassium gradient. So if we put a bunch of potassium on surrounding the stomata, what will happen? And so this leads us into activity 2, where we're going to be taking epidermal peels and observing the closure and opening of stomata using sucrose and ABA. And so there's basically 4 parts to this. The first part, so you'll make an epidermal peel. I guess we should go over how to do that. I'm just gonna make myself a little bigger. Okay, we have this geranium leaf here. The way that I like to make epidermal peels, or at least get a part of the epidermis to look at, is simply by ripping the leaf. And so I'll try to show you here. And so if you rip the leaf and then kind of pull the leaf, I'm just trying to show you, just pull the leaf apart, you are likely to pull off part of the epidermis. And so the epidermis is the white layer that you see that is surrounding the green. And so that's the skin per se of the plant. And so with tweezers, I don't have tweezers here, but let's see if we can do this. You can, yeah, you can grab that. I hope we can all see this. Grab that and peel a part of the epidermis off. And so I'll then take this piece, put it on a slide, drop water on it, and then you can put a cover cell over it, cover slip over that. And that's how we're gonna make the wet mount slide. So you just need a small piece. This is a pretty large piece. But yep, that's how you do it. Just put that down. Ripping is the easiest way that I like to do it, but you can also do it by using tweezers and kind of trying to peel that skin off, like I said. And so once you get that wet slide or that wet mount created with your epidermal peel, you're gonna basically do 4 washes of 4 different, 3 washes really, with 3 different substances to see what will open and close the stomata. And so in the first part, you'll observe the stomata just in the water. In the second part, you'll drop some sucrose around, or drop some sucrose on the slide to see what happens. You'll then in the third part wash the sucrose away with water and observe again what happens. And then finally you'll drop some ABA onto the slide and then observe what happens there. Okay, so that basically concludes the end of my explanation of the activities. So due in 2 weeks, we have pages 3 to 7 in your handout. And then we will be asking you to write a full lab report on activity 1. And so from here, I'm going to transition into some suggestions and basically just step you through the instructions that we have in the packet here. But it is very important to read this over the guidelines for the lab report. It basically steps you through how to format it and how to put it together. The one thing it doesn't do is go over how to make your figures and tables. And so if you need help with that, please let your TA know. All right, so I'm gonna move my little thing up here. Okay, so for the introduction, again, we've said this, or I've said this 1,000 times to my students, but you're gonna organize your introduction like a funnel. So you're gonna start with those broad ideas first. And so that is, what are stomata? What is their structure? And then lead into what their main function is for the plant and how do they work. That's all broad ideas that your reader needs to know to understand your experiment. And so here you can cite primary literature. And then after you introduce your reader to what your experiment is on and what you're going to be doing, or I'm sorry, not what you're going to be doing, what your research is on, you can lead into your specific research question and to the hypotheses you're going to do. And so for this, that is investigating the effects of the different treatments or the different treatments on transpiration rate. And so this is the light, the dark, the fan, and the AVA. You do not need to write this lab report for part 2. Part 2 is just for observation. So focus on part 1. And then so after you introduce to your reader your specific research questions and hypotheses, or your research question, you can lead into what your hypotheses are going to be. So what do you think will happen to transpiration rate when you use these different treatments? And so I also recommend to go back through your handouts while writing this, because we make you write hypotheses, and so you'll want to make sure you write the hypotheses in the way that we're asking you to. So make sure you look back to your lab handout as well. All right, so after the introduction, we have our methods. Some things that I see when people are writing their method section is that they tend to write it like a lab handout, where it's like a lab handout. It's more instructional rather than this is what I did. So don't write it like it's not we're not asking you to do a lab handout. We're asking you to just tell us in a paragraph form what you did to test the hypotheses you stated at the end of your hypothesis. And so you'll have to write methods on how you basically how you did the experiment. So you got this scintillation vials and you put the petioles in with parafilm, and then you place those 4 vials in different areas and then weighed them every 15 minutes to figure out how much water was being lost in the process. You'll also recount any of the statistical methods that you did. And so I think the only thing that we'll be doing is averaging our data. And so you want to say that in the methods as well. For the results, you're briefly going to describe the important points that you collected and how they compare to the other data that you collected. And so, so back to the method, sorry, one other point is we're going to give you the class data, so the class averages. And so you'll want to also put in the methods that, you know, your class did the experiment and these are the averages that you took over the course of the hour. Okay, oops, sorry. Okay, so back to results, sorry. So you're going to describe your important data points. So you can basically list the class averages, you can list your averages. And then this is the time to point out any outlying data points that you may have gotten, any weird data that doesn't make much sense. It's important to note that each time you describe a data point or a finding, you need to refer to the figure and table where it can be found. It's down here, but you need to include any tables and figures with proper captions in this results section. I prefer the captions or the tables and figures to be at the end. So I like to read my results before I look at the figures, but that's just my preference, I guess. You need to have a table and a figure. So that was one common mistake that I saw in some of the work that I was grading, is that you need to have a table and a figure. And so we have a figure here, and so this can be any sort of graph that you create out of the data. And then we also have a table. And so these are 2 different ways of visualizing information, and they are not interchangeable. And so one thing to also note is that figure captions go below the plot or the figure, and table captions go above the table. And then another important note is to just not interpret the data in this section. So that interpreting the data comes into play in the discussion. And so this is just one way to format your discussion, but if you're completely lost, I recommend taking this approach. And so in the first paragraph, you want to interpret your results in the context of your hypothesis. And so you'll start discussing the averages and their interpretation. So what do your results mean? And then you can note the outliers and maybe discuss that. The point of the discussion is to place your findings in context for the reader. So help the reader understand why you got the results that you did. In your second paragraph, you're going to bring it back to the broad idea. So what have you found out about which factors influence stomata, how stomata work? What does this mean for plants in a context more broad than your experiment? So this is where you can be bringing in literature citing other articles about how stomata work, et cetera, et cetera. And yes, again, add the background information from the primary literature to add the context. One other important note is that we're using APA formatting. Citing, and I guess one thing that I have noticed continually is that when people use Google Scholar or the library like actual citation generator, it doesn't give you the correct formatting. So don't trust it, check it. It's not that hard to take the information that they give you and change it into the correct formatting. And so you can see here, oops, this is something that I got off, I believe, of Google Scholar for the APA format. You can see that it has the first and last name. So for everyone. So that's not the format we want. We want last name and first initial. They have the title, which is fine. Journal of Experimental Botany, fine. But we don't ask for like the volume issue. You don't have to type that out. And so we want you to put it into the we want you to put it in the format that we're asking. So just make sure you can put the DOI if you want, but you don't have to. And then again, the date here, oh, I keep pressing, the date, May 2011, in APA format, the date goes after the authors. So just make sure you're checking it, make sure you're putting it in the correct format. And that is it. So if you guys have any questions, please let your TA know. Again, you'll have 2 weeks to complete the full lab report, and the handouts or this packet is also due at that time. Okay.